All right, good morning, everyone. Welcome to Virtual Breakfast. It's getting to be the end of the season for us, and we've got a good topic for you today. So Dr. Marisol Quintanilla, I'm told she won't worry about her name, but she is from Chile and is going to talk about monitoring nematode resistance in soybeans this morning.
All right. So today I'm going to be talking about soybean cyst nematode. Soybean cyst nematode is the #1 yield robbing pathogen in North America. An outline of what we're going to talk about, a short basic biology about soybean cyst nematode, and then some research finding from our lab and management of SCN, and some current and future research. So for soybean cyst nematode, the life cycle is what comes first, the chicken or the egg, right? So we'll go start with an egg. So when you have an egg there, the egg hatches and the juvenile, a worm, goes inside the root and it sets, it develops a special place for it to feed. Those cells become enlarged and fused and specialized to feed the nematode. The nematode becomes enlarged, sedentary. It doesn't have to move. It doesn't have to go anywhere to feed, at least the female. The male, yes, the male needs to move around in order to mate, in order to find females. But the female becomes sedentary and becomes full of egg, can lay an egg matrix, and can lay eggs directly onto the soil. And when it dies, when the female dies, it dies with full of eggs, 200 eggs, 150, 250 eggs. And that package with the eggs is called a cyst, and that can live like 15 years, 10 years, 15 years, 20 years. It can persist, waiting for you to plant the right crop in order for them to hatch and start the life cycle again. So once you have soybean cyst nematode, getting rid of it is very difficult because of how long-lived they are. You can see them with your naked eye. You can see the roots right here, and those little white dots are the soybean cyst nematode. They can look anywhere from white to yellow or tan, depending how old they are. When they're fresh and young, you know, they look white, and as they get aged, they start getting this tan color. So some of the symptoms are stunted growth, yellowing, early senescence, and they're all very generic. So you can't really tell by above ground cyst symptoms. You need to actually uproot the plants and look for those cysts in the roots and send soil samples to the MSU lab, the MSU plant diagnostic lab. Also, the nematodes make the plant more susceptible to certain plant pathogens like sudden death syndrome. So certain fungi, because they're injuring the root, moving in and out of the root. So here's a better picture. You can see the cyst directly on the roots, those little white things. So you can see that with your naked eye. You might need a magnifying glass to see this much clarity. Okay. So what are some of the best management practices? One is to rotate with non-host. So wheat, corn, just about every crop except legumes like beans. You know, beans is not a good idea to rotate with them like dry beans. But most other rotational crops, most vegetables are good rotation crops, most, and of course, wheat and corn. And wheat is especially good. We made a special discovery that we are very excited about. regarding wheat that we will inform you more as more research comes through. But we strongly recommend, if you have a severe problem with soybean cyst nematode, we strongly recommend that you rotate with non-host, and if you can, wheat, and also rotating resistant sources of resistance. So don't plant only one source of resistance. Most soybean cyst nematodes have developed, in Michigan, have developed resistance to P8788, the most common source of resistance. So rotating with the source of resistance as Peking is really good. Do not just switch to Peking. Use it in a rotation. So you can do 2 years P8788, 1 year Peking. Because if you switch only to Peking, then the nematodes will quickly develop resistance to Peking. And that, that'll defeat the purpose. So if you rotate with Peking and P8788, you get the highest yield and also the lowest reduction of SCN density, and that has been published. So we have researched this. There was a 4-year trial, and the results are clear and conclusive. Okay, we have done some cover crop trials. The main conclusion here: most cover crops are non-host and don't have much impact on soybean cyst nematode, besides just having bare soil, besides not having anything at all. You can see here, of course, the susceptible soybean is a good host. Everything else is not significantly different. Okay. We're continuing our cover crop work. And regarding manures, we have done several trials with manures and have found that certain manures are more effective than others for egg reduction. So in previous trials, we have found significant difference in like poultry manure, and there's also research from other labs showing significant difference with swine manure. So you can see there are differences here in poultry manure. Okay, I need to, my pictures here is blocking my, I need to make this smaller, I apologize. All right, so swine manure and poultry manure and layer ash blend, which is a product from Morgan Composting, all had lower numbers of, but the difference here were not significant, but in previous trials they were. All right, so another trial that we did is test again different sources of resistance, and we found that most soybean cyst nematode are able to reproduce very well in P8788. In fact, better even than the susceptible, but there's reduction there with Peking. So we do recommend that you rotate with Peking. We are doing further research in the greenhouse with different concentrations with manures in order to select the best manures for nematode control. And you can see here we're adding into containers different concentrations of manures and different types of manures, and we're trying to see about how do they affect on egg hatching, and we're comparing this with untreated control or compounds like that promote hatching or root exudate as a positive control. And you can see here that in a layer ash blend and layer manure, which are poultry compost or poultry manure, blends, or in layer ash blend has a couple of combinations, you can see it really inhibited egg hatching. There's mortality there too. Some of them have died. The high carbon and dairy manure did seem to promote egg hatching beyond root exudate. So that's something that we can explore, like when you're planting corn and you want to promote egg hatching, these kind of manures can be used to promote egg hatching, because if they hatch when there's no soybean, it's a suicide mission, and those numbers are reduced, significantly reduced. When you are going to plant soybean, there's other options that are actually preventing egg hatching and causing mortality. Unsurprising, all our previous research in several crops has shown that layer ash blend and chicken manure kill nematodes and prevent their hatching, while dairy manure and ones that are more plant-based, the high carbon, do not have much mortality to nematodes, don't kill them very well. But it was surprising to us that they promote the egg hatching beyond root exudate. So this can be a good game to play if you want to promote egg hatching when you're planting corn. There's certain manures that you can use that can promote egg hatching. And when you don't want them to hatch, there's certain manures that you can use that inhibit their hatching. So very nice results. Here is similar. Zinc chloride is a compound known to promote egg hatching. And we can see here that poultry manure really inhibiting, and swine manure. I mean, all of these were significant from zinc fluoride. From plain root exudate, poultry manure was significantly lower than root exudate. So this is a second trial confirming our previous results. All right, so we are doing also trials in microplots. We established some new beautiful microplots so that we can do research in environment that is more similar to what you're growing in, but also control nematode numbers, because we have trials in greenhouses, and sometimes the temperature conditions are very different from the field. So microplots are similar to your field conditions. All right, so we're also testing different bacteria that are naturally found in compost and manures to see if any of them are helping us control this nematode. We're doing a survey this fall. So if you're interested in being part of our survey, please send me an email. We're doing a survey in which we're sampling fields, and we have a questionnaire that we would like to for you to fill, and then we will take samples, and we will check if your nematodes are whether you have soybean cyst nematode or not. If you do have it, are they resistant to Peking? Are they resistant to P8788? And are there any management practices that you and others are using associated with higher nematode numbers, lower nematode numbers, etc.? Soil type, all of that we'll consider in the survey. So we are also exploring a question about wheat. Very interesting question. And I want to thank Michigan Soybean Promotion Committee. They have provided a lot of funding for us. The SCN Coalition, Soybean Cyst Nematode Coalition, is a good website where you can get detailed, clear information on soybean cyst nematode management. Again, SCN Coalition, Soybean Cyst Nematode Coalition. If you Google it, you'll find it very quickly. And Michelle also will share the website with you. And we are collaborating, and we are partners with them. USDA NIFA has provided funding for us. Project Green, Ned Berkey, Mike Staten, and of course, and Phil Katz and all the team here for inviting us. Eric, he has invited us to talk. Eric Anderson, he has invited us to talks in the field. If I keep on going, I'll keep on going forever with the thank yous. But a thank you to all and my team, my technicians, my students, Racie, who has done the work, my postdocs. So that's it. And if there's time for questions, I'm available for questions. And the email that you see on the screen is also the email that you can send if you're interested in the survey.
All right. Thank you for attending our virtual breakfast. Please remember that you can find us on Twitter and Apple Podcasts, Spotify. You can also follow us on Facebook or find us on our website. I think we're ready for questions now. Ira asked a question about the years between rotating sources of resistance, and Marisol did answer that in the questions, but I'd like her to go ahead and discuss that so everybody can hear it.
Oh yeah, well, I think the best thing, well, if you're planning to plant continuous soybean, which is something that most people would not recommend, including myself, if you're planning to do I would recommend 2 years of PI 8788 with 1 year of Peking. That is to protect the effectiveness of Peking, because Peking, my research, we did a 4-year trial, and our research, and I shared, if you look in the chat, I shared the paper, so there's a link to the paper, you can read the whole paper. Clearly demonstrates Peking effectiveness goes down with time. You get a huge increase, you get an increase in yield and decrease of nematode numbers the first time you plant Peking, and if you keep on planting Peking after that, it declines and it goes down even below acceptable. So no, I would not recommend replacing PIDX 788 with Peking, but it's a useful tool in rotation. So 2 years of PIDX 788, 1 year of Peking. The best thing is to rotate with non-host. Let's say you plant soybean, and then you plant corn, and then you plant soybean, or you can plant soybean and then you plant wheat, etc. But on the years that you plant soybean, still you want to rotate sources of resistance. And I still think that 2 years of PI 788 and 1 year of Peking is probably the best combination for having high yields, reducing your nematode numbers, and preserving that effectiveness of Peking. There was a question about cover crops, and regarding that question about cover crops, none of the cover crops we tested showed a significant reduction beyond no crops at all fallow, but a rotational crop, wheat, seems to do a magic number of them. We are trying to understand why, but yeah, wheat does have a beyond just a non-host.
All right, our next question is, how are the hatched eggs measured?
How are the hatched? Oh, very simple. Instead of being an egg, it's a worm. So if we put 100 eggs and they look like eggs, they're round, you know, and then then we look it up after we add whatever, either the compost or some special compounds to hatch them. Instead of having 100 eggs, now we have 20 worms, and the other 80 are eggs. Then we can say at 20% 20% hatched. That's just egg versus a worm. You know, worm looks long, you know, like a worm, and an egg, they're all rolled up in an egg, and it looks like an egg, you know, just like you can tell a chick versus, you know, a chick versus an egg.
All right. Are there differences within the sources of resistance in their ability to reduce egg hatching?
Yes, for sure. We have done trials on this. Some of these sources of resistance that are very effective at not being a host for soybean cyst nematode. Dr. Bird knows their name by memory. If it ends at 5.4, but anyway, that one that it's not really available for growers, but that's very effective. Also does not induce hatching, so it seems like it doesn't promote the hatching. While P-8788 and Peking do induce hatching on different levels, and it seems that Peking induces hatching more than all the others, even more than susceptible. So we are thinking, we were playing around with this. We're not recommending this to growers yet, just playing around. We were thinking of planting, let's say if planting soybeans for like a couple weeks, like 6 weeks, and then terminating them, herbiciding them, and see if that would induce a suicide mission. They are going to hatch, and they hatch, and then since the nematode is since the plant is terminated, they would die. So we did do that trial. So yes, different varieties do induce hatching at different levels. Yeah.
Marisol, on that question, I wanted to find out if there were differences within a source of resistance like PI 88788. Are there differences between varieties of soybeans in their ability to reduce the number of soybean cysts? So are there differences out there, and how do we decipher those differences?
Oh, there are differences. There are differences because the SCN resistance for PI8788 is kind of like the numbers of copies in a gene. So some varieties will be more resistant than others, even though they're all PI8788. They'll have more copies than others. But I cannot tell you this PI8788 variety is better than the other. There's people who do that. There's people who do variety trials, but I don't have them by memory. I can't start spitting out names of varieties that are better than others that are PI8788. Whichever variety, if it has PI8788, if your soybean cyst nematode has resistance to PI8788 or has broken through the resistance, it won't be as effective as a Peking, for example. So rotating sources of resistance is more powerful than just rotating varieties. But yes, not all, even though maybe varieties might be all PI8788, they don't all work the same against SCN. Some would be more effective than others. Did I answer your question, Phil, this time?
Yes, thank you.
Yeah.
All right, we're going to give you a little bit of a break, Marisol.
No problem.
We have a question for Jeff. Jeff, will Hurricane Lee block the high pressure over us now from moving off to the east?
And a very interesting question. And as I mentioned earlier, and I probably was a little too went a little too far, said very little impact or no impact on us here. But we know with increase, and it's not clear why, but we know that many times that hurricanes and other tropical cyclones around the world, this would include, of course, typhoons and other part of the Pacific, et cetera. But when those storms move into the mid-latitudes, many times they result in a deepening of the jet stream pattern. And this is something, there's a lot of research going on in this right now. It definitely happens in the Northern Pacific. We think it happens in the Atlantic as well. So while the answer, again, is probably very small impact on us here, it could be that we will see a little bit a stronger ridge develop over the eastern U.S. here, which is what we're all expecting next week and beyond. And that would, if that happens, that would mean a little bit warmer and probably dry. So more accentuated pattern with that ridge. So I think that there's a chance we could see that, although most of the impacts with Lee are going to be way off to our east and then ultimately into the North Atlantic and ultimately Europe and instead of here. But there's a chance there could be a small indirect effect, I think.
All right, thank you. Our next few questions are for Marisol again. Eric wants to know, what is the window after harvest that is optimum for soil sampling soil for soybean cyst nematode after specific protocols or any specific protocols to keep in mind when sampling, for example, between the rows or within the rows?
At harvest is a great time. Right after harvest is a great time. Nematodes are not, they don't move very fast, you know, they don't travel rapidly. So you don't have to get there immediately the day after harvest. Even if you sample next spring, you're still gonna find soybean cyst nematode. Obviously, right after harvest, you would find the highest population. In the row or between the row, in the row where the roots are is where you're going to find the highest population. So if your objective is you want to make sure you know if you have soybean cyst nematode and you want to catch them if they're there, then sample in the root zone where the roots are. Yeah.
Okay. Mark would like to know, do you expect new sources of soybean cyst nematode resistance from new technologies such as CRISPR or CRISPR, yep, or genetic modification?
Yes, there are companies working on this. There are scientists working on this full time in different universities. Yes, I can't tell you the exact timeline because I'm not working on that. My work is more applied and, you know, checking on management practices. But yes, there are new sources of resistance. People working on this, and sooner or later, we will get new ones. And as time goes by, hopefully we will get several new sources of resistance. And when we do, it would be great to include them in the rotation. So nature is just the way nature is. You know, things break through resistance. If you apply the same herbicide on the weeds for 20 years, eventually those weeds will not die with that herbicide. You're selecting for resistance. So nematodes are no different in this sense. If you plant the same source of resistance for 20 years, nematodes will eventually be able to reproduce in that source of resistance because you're selecting for resistance. So rotating keeps them guessing. Just like you need to rotate different types of herbicides with different modes of action, and you need to rotate different insecticides with different modes of action, it is best to rotate different sources of resistance for soybean cyst nematode also in order to preserve the effectiveness of that genetic material or that source of resistance. Yes, so I do expect to have different kinds coming.
Great. And I think our final question, unless anybody wants to include one more, is what about planting both types of resistant soybeans together?
Yeah, I mean, surely that can be done. I have not done it. You would get, you know, you would get more of a mixed population, and it will kind of delay this resistance building. Still would be a little bit complicated. You need to get the right maturity group and the right everything. I think alternating by year might be easier and even more effective in the sense that if you plant 2 years PIDEX 788, those nematodes are developing resistance to PIDEX 788, and then you break it with Peking, and you get a yield bump because those nematodes are, you know, disrupted in their development of resistance, and then I think it's it will increase your yield maybe further, and it's easier to manage. You don't have to worry about maturity groups and making sure they match and the right development and the right heights. If they have differences in whatever, I think it would be easier to just have one at a time.
Michelle, this is Mike Staton.
Go ahead, Mike.
And good morning. I just wanted to mention that there's an article on MSUE News, and it's going to be talking about some harvest considerations in soybeans in fields of soybeans that have variable maturity. It's a situation we're going to see this year more than usual because of the delayed emergence that we had of any of the fields that were planted into dry soil. We had emergence within a given field vary by, anywhere from 4 to 6 weeks. So there's just some recommendations on how to deal with that at harvest time in an article that has been submitted to MSUE News. This article was also previously published in the Michigan Soybean News. So growers that receive that, look for that article in there as well. Thanks.
Great. Thank you, Mike. And I see Erin would like to talk.
Thank you. This is Erin Hill from the Diagnostic Lab. And I just wanted to put out there a reminder that this is a good time to be collecting weed seeds if you want to have them screened for herbicide resistance over the fall and winter months. We will be reposting an article about this very soon on MSUE News, so we'll share that next week. But this is the time to be collecting. You need to collect from at least 5 plants of your suspected resistant species. And you can submit those on our website. There's the form and all the written down information about what species are covered by the Michigan Soybean Committee. Those would be the pigweeds, common lamb's quarters, mare's tail, and the ragweed species. But we can screen other species as well. If it's something outside of those species, then I just ask that you email me or contact me so we can make sure that we have a control set of seeds that we can use for that screening. So be collecting those. We ask that the seeds be turned into the lab by mid-November so that we can get those screens done in a timely manner before you need to make decisions for next year. Thanks.
Thank you. I think Jeff left. Mark had a question about the wildfires. Mark, I'll get you Jeff's email and you can ask that to him directly. Marisol made a note to mention that when you do send your soil to the diagnostic lab for soybean cyst nematode sampling, it is covered by checkoff dollars. So don't hesitate to do that if you need to know that information.
Michelle, what we can do is we can ask Jeff to make sure and go over that question that Mark had as far as the smoke next week. I think he's probably going to have a little extended forecast and maybe he can talk about that next week. So we'll make sure and get that to him, Mark.
Thank you. Well, I don't see any other specialists raising their hands and I don't see any other questions. So everybody have a great week and we look forward to seeing you next week.